And this is the last bit, and this is just going to look at tissue repair. And once we're finished with tissue repair, we are done with tissues in Chapter 5. So tissue can be repaired in two different ways. Regeneration, and that's the replacement of dull or damaged cells by the same type of cell as before. Restores normal function. Skin injuries, liver cells, they usually regenerate. Another one is fibrosis. And you can also have fibrosis on skin. We're going to kind of look at these two different types, but fibrosis, replacement of cells with scar tissue. So if you ever cut yourself really bad that you had a scar and that scar didn't go away, that is going to force the skin or the organ to hold together, but it does not restore normal function. Severe cuts, burns, healing of muscle injuries, scarring of lungs by tuberculosis, these all produces fibrosis. And if you think about scarring of the lungs, if you don't have that elasticity of the lungs to open and close to allow air in and air out, if you had a severe case of tuberculosis, you aren't able after that disease to have the air transportation from outside to inside of the body. So the oxygen can't get into the blood as easily. So people who've had tuberculosis usually are going to have long-term effects from that. But we'll kind of look at this. And I mentioned, skin. If you have a scar, touch it, kind of move it around. You can see the skin around it all moves in like one way or another when you put different types of stresses on it. But that fibrosis is pretty much just holding one side together from another side. So regeneration, normal function. Fibrosis, just to hold them together until hopefully cells can grow underneath them and then become fully regenerated. So let's look at how this actually happens. First, healing of a cut in the skin, because it's the most likely one to happen. So what happens is you slice between the different tissues, and in this case, the skin. So blood vessels bleed into the cut. This causes mast cells and damaged cells to release histamine. Histamine dilates the blood vessel, and dilation will increase blood flow to the area, and you're like, oh my gosh, so histamine is actually going to create more bleeding? That's true. It's also going to make capillaries more permeable, so more able to bring stuff in. And pretty much what happens the first cut of bleeding, it's going to create more antibodies into that area. Antibodies will stop viruses and bacteria. It'll also bring more clotting proteins. Clotting proteins are good because that's what's going to stop the bleeding out. And blood cells to go in there and start to form in that clotting area. Also, the white blood cells to clean up any debris and also stop infection. So first thing that happens is it forms a clot. So all of that clotting stuff starts to form, and then those fibrin or those fibers start to loosely knit the cut together. This stops pathogens from growing around and through different parts of the site, but also it starts to have these red blood cells get caught in them. And there's these red blood cells that start to form the scab. And once the scab is there, it seals the wound and blocks further infections. Then macrophages move in there and they phagocytize tissue debris and also phagocytize bacteria and viruses that might have come in when it was bleeding. Then after that is done, what happens is, and you can see it in this picture, this is a lot of different things, but you'll see here, you'll have all of these different types of clotting. It starts growing from the bottom and up. You have blood vessels that start to regrow in there. You have all of these different fibrous things right here, and you have the scab underneath it. But you have macrophages that grow in there to start dissolving all that fibrin, start dissolving some of that fibrosis that formed, and start to clear the way for a new layer of the stratum basale to be there, and then the stratum basale can then start growing up. That's why when your scab first falls out, the skin looks a little weird. And the skin looks a little weird because you still have new cells growing up underneath it. And eventually, the macrophages remove the blood clot. So then all this that's happening, the connective tissue undergoes fibrosis, which we talked about. That was on the other slide. But here you can see the stratum basale is back there, and it starts to grow up and up and up through the spinosum, granulosum, and then this is not thick skin, so no stratum lucidum, but it does have a stratum corneum, and eventually it will start to remodel. It takes several weeks after the injury, may take up to 2 years depending on how deep that cut is, and what remodeling is doing, it's now going to start to remodel all the area underneath it. And like I said, depending on how bad it is, if you just cut yourself with a paper cut or something, you might not even see it, but if you have a bigger cut, it might take about 4 to 5 weeks, or if you had a really bad injury, it can take 2 years, too many years after that. And what's happening with this? Atrophy. The term atrophy, the shrinking of tissue through loss of cell size. So you can have different types of atrophy. If you've ever had a broken bone and you had it in a cast, you'll see all of your muscles start to atrophy or die because you're not using your arm anymore. And this happens to astronauts in space because they don't use them because there's no gravity. You can also have something called necrosis. And this is premature pathological death of tissue due to trauma, toxins, or infection. Also, necrosis can happen if you don't have oxygen. That's going to be a specific type that causes hypoxia, but any type of thing that happens where you don't have the proper nutrients going to a cell can cause cell death. And infarction is when the blood supply is cut off. You might have had a myocardial infarction or a heart attack. That's when you don't have the blood supply to the cells of the heart. That's called an infarction. Also, gangrene is tissue necrosis due to insufficient blood supply, or you could have diabetes, which can cause gangrene. You can have an ulcer, which is bed sores or pressure sores because the blood flow doesn't flow around. And then also gas, which is anaerobic bacterial infection in your digestive system. So if you don't have the correct blood supply, then you can have different types of necrosis. Apoptosis is programmed cell death. We mentioned that in cell. They go through this every so often. Different tissues have different types. But the most important thing is apoptosis is good. And we talked about phagocytized by macrophages and other cells to start to destroy them. And then billions of cells die by apoptosis. And every cell has a built-in suicide program except for cancer cells. And then you have tissue engineering. And this is the artificial production of tissue and organs in the lab for implantation in human body. The framework of collagen and biodegradable polyester fibers, where you have different types of skin graft, depending on what it is. They grow this in the lab, and they just peel it off, put it on your skin, and those collagen fibers, because they're grown in the lab, they're not going to be rejected by your body, and they can stop blood, or they can be used for burns and cuts. They use human cells. Some of them can be grown in a bioreactor inside of a mouse. We'll kind of look at this. And the reason why we kind of looked at how cells and tissue grow is because they still do this today, where they will take human tissue, those human seed that we talked about, and they put it into a bio model. They use mice, they use rats. And what happens is, if people can put in collagen cells from a person's ear, maybe that person lost it in a fire. Maybe they lost it in a car accident. Maybe they lost it, in some other horrific way. But what they can do is they grow the actual person's ear on a mouse or a rat. And this is a nude mouse or a nude rat, which means it doesn't have hair. Then they just grow it on the back of it. They cut this off, and then they can put that back on the human person. And then through plastic surgery, they reconnect blood supply and everything else. They do that via leeches still and other types of ways to produce pressure so the blood goes into this tissue so it doesn't die. But what it does is you take the actual human DNA, grow it in the back of an animal model, and can actually create these different things. They do this for heart valves, coronary arteries, bone, liver, and tendons. Sometimes for heart surgeries, they still use your own either arteries or veins. But for the most part, for most plastic surgeries, noses, ears, and stuff like that, they're still growing them on the back of a rat. So that concludes this lecture on tissue. Good luck.